I'm Dr. Dmitro Klokal by European Wellness Academy with title Xenogenic Cell Therapy. So what makes the fetal precursor stem cells the ideal option for tissue regeneration? First of all, this type of cells, this type of stem cell, are being committed to the predetermined path of differentiation along a single cellular lineage only. Let's look at the example of the cardiac regeneration again. As we have already discussed, non-progenitor stem cells, the MSCs, and other pluripotent stem cells perform their effect predominantly via the pericrine effect that does promote angiogenesis, improve host cell metabolism, activate endogenous pool of stem cells to a certain extent, though as it turns out, quite limited. It also has the anti-inflammatory effect, and as we all know, chronic low-grade inflammation is one of the main hallmarks of the age-related diseases. They also have an immunomodulatory effect via expression of the tissue transforming factor, TNF, interleukin 6 and 10, and interferon. In addition to all of that, fetal precursor progenitor stem cells do what other stem cells fail to do. They perform homing effect, produce a new cardiomyocytes formation, stimulate and perform cardiac remodeling, thus improve the contractility and produce the actual cardiac repair. Fetal precursor stem cells fulfill all the criteria of the stem cell transplantation and successful tissue regeneration, such as organ specificity. Cells of the same organs belonging to different species carry lots of similarities, especially between the corresponding cells of identical organs or tissue of various animal species, which, including in humans, are biologically similar. Interestingly, that these cells, being in the fetal stage, carry a considerably less immunogenicity compared to the adult stem cells. That allows to avoid immunosuppression. Next one is a homing effect. Implanted clusters of particular fetal progenitor cells of a specific organ or tissue from the donor disappear from the host implantation site within 48 to 72 hours, and from 25 to 75% of implanted stem cells incorporate within 5 to 7 days into the corresponding homing organ or tissue of the host. Next criteria or next principle is the principle of homology. The contemporary medicine is operating on the molecular levels. Proteins and other biologically active molecules from various organisms of different species are homologous to each other. It means that similarity is significant over the entire amino acid sequence. Homologous molecules carry out similar functions. The studies of the homing effect started over 50 years ago. Initially, homing principle was proven empirically. Thus, Dr. Gerhard Hofecker, the MD, and Hans Niedelmüller from Ludwig Boltzmann Institute of Experimental Gerontology of the University of Veterinary Medicine in Vienna, Austria, fed young rats with radioactive amino acids so that all the proteins in the body became labeled with radioactivity. They then injected cellular extracts from these animals' livers, hearts, and kidneys into recipient animals. 2 years later, they found radioactive traces of the cellular material in the corresponding organs of the recipient animals. Liver went to liver, heart cells, proteins went to heart, and kidney molecules went to kidney. Dr. Kment from Vienna University found that fetal cells actually shifted the biological age markers in all rats, causing significant improvements in the skin, aorta, liver, kidney, and heart, as well as in motor activity and learning ability of the animals. In subsequent studies on 700 male rats, Dr. Niedelmüller, Hofecker, and Skalitsky found that fetal extracts prepared from either sheep testes synthesis of mesenchyme tissue, which is a very early connective tissue cells, transformed fundamental aspects of the aging process. Those included increased DNA repair, decreased DNA damage, improved maximum oxygen uptake, and the shifting of the spontaneous activity in all the animals towards that of younger ones. Most importantly, the Austrian researchers found that the fetal extracts increased the mean and maximum life expectancy in comparison to the controls. Dr. Harbers found that the coefficient of reabsorption in injected cellular clusters of liver cells, which is normally from 5 to 7% per day, increased to 15 to 20% in a liver after it has been pre-damaged. Dr. Thomas Starzil has done a remarkable work on demonstrating the thymic regeneration with xenogenic thymic cell transplants, and also demonstrated a direct homing effect of the fetal thymic progenitor stem cell. And of course, we have Professor Günther Blobel, who has performed and proven and explained the homing principle on the cellular and molecular levels. He proved that and discovered and proved that proteins have intrinsic signals that govern their transport and localization in the cells. I'm Dr. Dmitro Klokal by European Wellness Academy with title Xenogenic Cell Therapy. Other unique properties of fetal precursor stem cells are high level of readiness to differentiate and to undergo changes, especially in response to environmental stimuli, in accordance to recipients' epigenome, and also able to preserve their own epigenome throughout few generations. From the first year of medical school, we all remember that it is the microenvironment of the host, the recipient, that influences the donor cells, and there are no such biological mechanisms that allow the transfer of the genome from the donor cells to the recipients. Next one, easy adaptability due to the plasticity of tissues, which gradually decreases with each successive fetal stage and almost disappears at the end of development. The next one is a high potential for growth, migration, ability to create cell-to-cell contacts and cellular interactions. The next unique property of fetal precursor stem cells is highest frequency and speed of cell division and proliferation. Number 4, highest production of various biological substances such as growth factors and etc., which facilitate survival of cells after implantation and stimulate damaged tissues and organs of the host. Number 5, low immunogenicity with consequently much weaker immune response of the host as compared with the implantation of the cells of the adult origin. And last but not the least, capability of survival on lesser amount of oxygen and nutrients and on energy supplied by glycolysis only. How are the stem cells, these particular stem cells, prepared? They are prepared using a patented method of cell culturing called as the primary tissue culture. The process is conducted in a research-grade laboratory in completely aseptic conditions, when within 11 to 14 days the required type of fetal cells are cultured with all the necessary viability test, endotoxin test, etc. done throughout the entire process of preparation and culturing.